So Year 11s, today we're going to be looking at start of chapter 1.5, Introduction to Cell Membranes. In this few lectures we're going to be looking at, in this lecture, we're going to be looking at the structure and function of cell membranes. Firstly, we're going to be looking at the function. So in eukaryotic cells, we have two different membranes. We have the plasma membrane and the non-plasma membrane, and each one of those has a slightly different role. In the plasma membrane, that's the boundary around the cell. So this is our plasma membrane, or we can also call it our cell membrane. The non-plasma membrane is the membrane that goes into making up all of our different organelles. That endomembrane system that we've been talking about. So the plasma membrane surrounds the entire cell. It envelops the cytoplasm. The non-plasma membrane is what wraps around the organelles. Certainly the endomembrane system is the largest network of that non-plasma membrane. Prokaryotes, on the other hand, because they don't have any organelles or membrane-bound organelles, that means they don't have any non-plasma membrane. So they only have a plasma membrane or a cell membrane. There are some very small differences outside of the function. So the plasma membrane, we're going to go into quite a bit of detail about the function of that. The non-plasma membranes, we've already talked about the functions of those. That's when we've talked about the organelles. So we're going to go into some detail about the cell membrane, the plasma membrane. Non-plasma membranes have some small functional structural differences, compared to plasma membranes. The main one being their thickness. So non-plasma membranes, they're about 5 to 7 nanometers thick, and plasma membranes are between 7 to 9 nanometers thick. So there's a small difference in thickness, and that accounts for, I suppose, an increased strength of resistance to invasion, to being able to enter into these plasma membranes. The key role of all plasma membranes is to control entry, to stop certain substances from getting in and to prevent other substances from getting out. And the way that the plasma membrane does this is by being dotted with lots of different types of molecules. So we have some carbohydrates, some sugars, we have some proteins, and we have some fats. And this makes it selectively permeable. So the proteins themselves, they're able to monitor, and the glycoproteins, or the proteins that have sugars attached, they're able to monitor and control the entry of certain substances into the cell directly. But there are also the fats and the nature of the membrane itself, which also helps to restrict the amount of, or the type of substances that can come in. So we can monitor the entrance of hormones, we can make sure that other types of cells aren't able to fuse and enter into the cell, so bacterial cells and those sorts of things. So these proteins around the edge, these glycoproteins, they also help monitor this. So we'll draw these in. We'll start in red with our proteins, and they're all throughout the cell membrane. We also have our lipids, so our fatty molecules, however we want to draw those. We should be labeling those as well, so we'll quickly label those. So those are our lipids or fats. And the red were our proteins. And then alongside that, we're going to have some sugars. So, carbohydrates. And sometimes we're going to have a couple of proteins that have our sugars attached as well. So these are our glycoproteins. And they're there to monitor entry and exit into and out of the cell. So they're very, very critical structural features of the cell membrane. The other thing which we've learnt in with our organelles is that membranes can also increase reaction rate. So by certain cells being able to fold their membranes, or by certain organelles like our mitochondria and chloroplasts being able to fold their membranes, they're able to be covered with more proteins or more enzymes, so they're going to be able to facilitate these reactions to go far faster. The outer membrane for our cells, so now we're looking at plasma membranes, the outer membrane also helps to form an anchor point with the internal structures, with the cytoskeleton. So within this cell we're going to have lots of these skeletal structures, these cytoskeletal structures coming in and fusing together to form some rigidity. The cell membrane forms a key part in the structure of this. So you can see really now how important these different structures are and how complex cells can get very, very quickly. Pardon me. So, very quickly, our summary of membrane functions. And we're going to go into all of the different details, particularly about exchange, in much more detail in later slides. But to summarize membrane functions, they control entry and exit of molecules from the cell. They control essential proteins or essential reactions. So when we're looking at non-plasma membranes in the chloroplasts and mitochondria, critical in that, they act as barriers between the environment, between the extracellular fluid and the cellular components, between this extracellular fluid and cytoplasm. They restrict movement of substances within the cell, so they stop certain things from escaping the cell when they shouldn't, just by wandering out. The communication with cells via receptors. So these glycoproteins and proteins and lipids can actually detect other cells and communicate from that. And then lastly, production of electrical activity in excitatory cells. So if this were a nerve cell, then the cell membrane would actually be involved in creating that electrical current. A bit beyond the scope of our course, honestly. We're not going to cover any types of nerve conduction, but it is one of the most important roles of our cell membranes. Now, we've got up here our diagram, and you can see how crowded the cell membrane is. We've got these proteins in red, these lipids or fats. The lipids and fats are interchangeable. So these lipids in green, and these black carbohydrates, and then sometimes we've got glycoproteins with proteins and carbohydrates attached. So you can see there's this incredible complexity to the cell membrane. We give that complexity a term. So the current theory explaining the composition of the cell membrane is the fluid mosaic model. And it's basically summarized as that the membrane is fluid as all particles are able to move. So although the cytoskeleton helps anchor things in place, every single molecule within the plasma membrane is able to shift about, and its mosaic because it's composed of many different molecules. So if we were to try to track one of these proteins, let's say, we would see that over the lifetime of this cell, the protein might be starting here, and it would shift up to the top and then move all the way around and eventually come back into its original place, purely just by chance. This is a very fluid structure. It's also a mosaic. It is just interspersed with hundreds and millions of different types of proteins and thousands of different carbohydrates and hundreds of different lipids. There is just a terrific variety of different chemicals associated with the plasma membrane. This also leads it to be very, very effective at its role. If we look at all of those functions and we take just a couple, so that communication between different cells, the fact that it's able to be covered with so many different types of molecules means that it is more likely to be able to interact with more cells. So that it's going to be able to detect when it's a bacterial cell, because that's a cell that it's not familiar with, or that it's going to be able to detect a muscle cell very effectively. And if it's another muscle cell, it might want to attach itself to that muscle cell to form muscle tissue. So this is our general description. What we also need to consider before we finish up this lesson is what are the actual smallest parts. So if we consider something like DNA, which we looked at in year 10, DNA is made up of these nucleotides. DNA is a polymer, it's a massive, long, complex molecule, and the nucleotides are the monomers. Now our cell membrane is a massive chemical structure made up of lots of different kinds of chemicals, this mosaic idea, but it's got one feature that is by far the most dominant in the landscape of the cell membrane, and that is the phospholipid. Now we're going to have to get rid of this diagram in a moment, but I want to leave it up for just a little bit longer. The phospholipids are the blue parts of the cell. So the phospholipids, you can see, I mean, my diagram here is an indication of what the cell membrane actually looks like. These blue sections here represent the phospholipid, and they are the dominant feature of these cells. They are, as their name suggests, they are a lipid, so they're a fat. So our cell membranes are a fatty membrane. But they also have quite a unique feature. So fats have a common chemical structure. So they have a glycerol backbone. They have a glycerol backbone, and then they have these fatty acid chains. With our phospholipid though, instead of having 3 fatty acid chains, it has a phosphorylated alcohol. So it has a phosphate group attached to it. This is our phospholipid. Now this changes the nature of the substance. Ordinarily, lipids are uncharged. They are non-polar molecules. The phospholipid, because of that phosphate group, because of that alcohol, it actually becomes charged. And we can see that in the way that it behaves. There is a part of this structure of the phospholipid that dissolves in water. So we call it hydrophilic. And over here, the fatty acid chains do not like water at all. So they are hydrophobic. So we have this water-loving side and this water-hating side. This creates a real interesting relationship between these two different sides. And it means that the cell membrane acts as a very effective barrier. And we'll talk a lot more about this when we get into diffusion and osmosis and all of those sorts of things. But it acts as a very, very efficient barrier between the water in the extracellular fluid and the water in the cytoplasm and these fatty acid chains. This means that the phospholipid bilayer, the cell membrane, is impermeable to substances dissolved in water. Water itself is small enough to pass through, but it is impermeable to substances dissolved in water, to water-soluble substances. What this ends up meaning, so this is our general diagram of our phospholipid. Now I use this term that the cell membrane is a phospholipid bilayer. So if we simplify our diagram here, so each of these just represents a phospholipid. We're just going to draw a section of the cell membrane. So this is our phospholipid bilayer. This is the basic underlying structure of our cell membrane. And on this side is is our ECF. On this side is our cytoplasm. Now, both of these liquids are mostly water. So the hydrophilic heads, which I've symbolized with these circles, are attracted to the water, and the fatty acid chains are repelled by the water. So whenever we have a phospholipid, it is going to create a bubble between the fatty acid chains and the water that's surrounding it. And so this automatically forms, these phospholipid bilayers automatically form into these bubbles, or we call them micelles. Now that brings us to the end of this particular lesson. There's a lot more detail to go through with it, and we're going to get to that when we start to talk about exchange.